thank you all for joining us for the January 2024 edition of the Arima webinar series. We're going to be talking about structural variant detection using the Arima Bioinformatics Platform. We have over 400 registrants for this webinar, which clearly shows just how popular bioinformatics topics are. And you guys are in for a real treat because we have two fantastic Arima speakers presenting today. And with that, I'm going to hand it off to Sophia to give us an introduction to structural variant detection. Sophia, thanks so much for being here, and the floor is yours.
Thank you, Janetta, for the sweet introduction and for always inviting me and giving me the opportunity to get out of my little shell and present to the world. I'm very excited to be here. So welcome, everybody. Today, we're going to be talking about structural variant analysis in cancer, and we're going to be focusing on the Arima Bioinformatics Platform. So let's start with some introduction to our technology and why we believe that 3D genomics is great for research and for getting a better insight into the organization of the genome in the nucleus. So traditionally, linear genomics focuses on the sequence of the genome, but we're missing on the fact that the genome is not just a linear structure. It's actually folded in many different conformations inside the nucleus. And that's where the 3D genomics analysis comes in. They're giving you the opportunity, it gives you the opportunity to understand not only the sequence, but also the structure of the genome. So the way we look at it at Arima is that 3D genomics sits in the intersection of these three very important aspects of genomic analysis. We have the sequence. So the 3D genomics gives you the DNA sequence information on top of everything else because it's based on next generation sequencing. It also gives you information on the structure of the genome. And specifically for this webinar, we're going to be focusing on the ability to identify structural variants, which can be very important as cancer drivers and also in other types of disease. And it also gives you information on the specific regulation of the genes behind the 3D interactions that are established between genes and promoters, the gene promoters and enhancer elements, for example. So there are a lot of applications of 3D genomics. Some of you I hear potentially are from the assembly world. We have a lot of applications on understanding genomic organization and function. This is where our September webinar focused on. It gives you an insight into non-coding disease risk variants and what genes they're targeting and potential disease pathogenesis. It helps us discover the mechanisms of gene regulation. And for today, it's very important and helpful in detecting structural variants and gene fusions. So this is what we're going to be focusing today in this webinar. And let's talk about the importance of SVs, which stands for structural variants in cancer. So structural variants play a very significant role in cancer. More than 95% of cancers have one or more somatic structural variants, and about 30% of them have a known pathogenic structural variant that is used in diagnosis and for patient stratification, and it oftentimes can also guide the therapy decisions. That's why it's very important to be able to identify structural variants in a patient so that we can help them potentially receive better care. And of course, SVs alter the structure of the chromatin, and therefore they can potentially introduce new interactions with cancer genes through D loops and interactions that were not established before. So detection of structural variants is very important to us and for the clinical world because they can help with helping us understand disease mechanisms, potential diagnosis, and help us stratify our patients. So at Arima, we're focusing on an integrative approach, and there are a lot of, let's say, mutations in cancer genomes, about 4 to 5 driver mutations, both in coding and non-coding regions. And something that might be oftentimes neglected in this field is the non-coding element of potential structural variants. And also, not all cancers have known drivers that come from either a specific gene or a gene fusion. So we do really think that 3D genomics can help us identify both known and unknown genetic and epigenetic drivers in cancer genomes with a specific focus in the non-coding regions. So if we have for you a comparison of the technology landscape that is used to identify SVs. We have Hi-C, whole genome sequencing, targeted RNA, optical approaches, OGM, and also FISH and HiFi, which are some technologies used in the field. But we do think that using Hi-C to identify structural variants has some advantages over those. For example, Hi-C can help you identify fusions with non-coding regions of the genome, which, let's say, targeted RNA cannot do. Hi-C can help let's say, break down very complex structural variants that are not just one deletion. They comprise, let's say, of a translocation, deletions, and inversions all together, which very often happens in cancer genomes because we have chromothripsis and a lot of rearrangements happening. And that's something that FISH, for example, cannot identify. And a very important advantage of our technology is that it can actually be applied on FFP tissue that can be 10, 15, 20 years old. And that is not something that can be done in the other methods. Whole genome sequencing can potentially accommodate it if you have enough number of reads. But we do think that overall Hi-C has a lot of advantages, and it's worth exploring as a method to identify structural variants. So we think 3D genomics for structural variant detection can help us detect known and novel gene fusions. So still you're maintaining the capacity to identify fusions between genes, but also we're adding the benefit of identifying fusions that are missed by other technologies, such as the ones with the non-coding regions. And it can also be applied on FFP specimens. So we do think that these are a lot of advantages that, might convince you to consider doing some Hi-C. So how does it work? How do we detect gene fusions in 3D genomics, and how is the fusion manifested in our data? So let's say we have a normal cell where we have chromosome A and chromosome B that are not fused, right? And we get reads and interactions within each chromosome separately. We can see that the interactions here are contained within chromosome A, and gene A is not interacting with gene B in any way. But if we have a fusion that leads to a connection of these two chromosomes that previously were independent, we're starting to see reads that are spanning this breakpoint. The breakpoint is the area where the chromosomes have been cut off and reconnected with each other. So now with our 3D Hi-C analysis, we're going to be seeing reads that are coming from across this breakpoint. So part of the read will be aligning to chromosome A, part of the read will be aligning in chromosome B, and that is an indication of potential fusion there. And we don't have one cell where this happens. Usually we have a multitude of cells. So we're going to be having a lot of reads that are coming and from this particular fusion and that are spanning the breakpoint. And this is how Hi-C is able to identify structural variants. This is the basic idea behind it. So how does the Hi-C data being usually visualized? It's through the heat map. So for the newbies here, I think half of you have not worked with Hi-C before, but you've probably seen a heat map on a paper. So this is basically the entire genome in the form of a heat map. You have every chromosome organized here in this particular case by, I think, alphabetical order. And what you're going to see is that basically on a heat map, the more red the color, the higher the frequency of interactions in your cell population. So here, this is a normal cell. It doesn't have any structural variants, any translocations between chromosomes, right? So what you're seeing is that the majority of the signal is comprised within each chromosome in these little squares. And this is what we're representing here. With the same schematic as before, interactions are basically contained within each chromosome. Now you are going to be getting a background of interactions between chromosomes, but that in this case, it does not indicate any specific translocations. And if we look at how an abnormal karyotype looks like, you're going to start seeing interactions in the interchromosomal space, right? Here we do have an enrichment of signal between chromosomes, I guess 4 and 6, and that potentially indicates a structural variant. And this is how we would be able to identify that with these reads spanning the two chromosomes. So we have a very interesting case that we worked on here at Arima in collaboration with Matthias Nudrel from NYU Langone Health. We're very proud of this case, and I'm going to show you why detecting structural variants with Hi-C was very important in this case. So there was the case of a 15-year-old female who presented with headaches and seizures, and after resection, they run every single test that they had at NYU to identify fusions and copy number variations, and nothing came up as a positive hit for this particular tumor. The tumor recurred 6 months later. The diagnosis this time was glioblastoma, and the available methods in the clinic just showed a lot of complex genomic events on chromosome 9, but no specific driver was uncovered. So we worked with Matthias Nudrel, and we run our Hi-C method on this patient, and we were able to find an rearrangement on chromosome 9. So that's this is chromosome 9. If you zoom into this area, you're going to see some signal that is not there normally. I also want to mention that, of course, rearrangements can happen within the same chromosome. In the previous slide, we talked about interchromosomal cases, but in the intrachromosomal space, you can also have similar rearrangements occurring. And in this case, if you can see, these lines indicate our breakpoints. So you can appreciate that one of the breakpoints is very close to PDL1, but it's not in the gene. That's why it was missed by other methods. And the other end of the breakpoint is in a non-coding region. So there's no way an RNA-based approach could have identified that. So there is a rearrangement close to PDL1, and it was confirmed with immunohistochemistry that there was a overexpression of this protein in this patient, and there was a shift in the therapy. The patient was given pembrolizumab, and it was successful. The disease is stable. So this is an example of how methods such as Hi-C that can identify fusions between non-coding regions can be very useful in some cases. So at Arima, we have put together the Arima SV pipeline. It's a pipeline that's available on our GitHub. Some of you may have used it. It can help you start from FASTQ files and go all the way to your structural variant calls. So I'm just going to give a very brief overview of the tools we've chosen for this particular pipeline. We have Hiccup for alignment and filtering out the non-informative reads, which is a very well-established tool in the field. We are using Juicer to generate the Hi-C heat map, also a very well-supported tool, and Hi-C break finder for SV calling. The reason we chose these tools is because they're publicly available, they're well-supported, and we have packaged them in a way where it can be easily installed and executed. But for somebody who's not a bioinformatician, we wanted to take it one step further, and this is where I'm gonna give the floor to Ibrahim to introduce us to the Arima Bioinformatics Platform. Take it away.
Thank you, Sophia. That was an excellent introduction. And I am going to go ahead and share my version of the screen here. So as Sophia mentioned, the list of tools that we have were formed into the Arima SV pipeline. And in order to make this pipeline more accessible to our users, we have partnered with base pair biotech to launch the Arima Bioinformatics Platform. So what is this platform? It's a online user-friendly bioinformatics platform for targeting, our typical bench scientists. So you don't have to be a well-versed bioinformatician to be able to use this platform. It's designed exclusively for Arima customers for use with Arima kits, and it streamlines the process of analysis for non-computational researchers. And when we launched this pipeline specifically with the Arima SV pipeline first, so there's two pipelines or workflows that are available on the platform today. The SVQC pipeline, which is a quality control pipeline specific for determining whether your sample has the quality metrics that are suitable for structural variant analysis. It takes a shallow-seq sequencing run, so it could be a downsampled version of a deep sequencing Hi-C data set, or it could be just a shallow-seq run of 0.5 to 5 million reads. This will be free to all Arima Hi-C customers. So if you've purchased a Hi-C Plus kit, a Hi-C plus FFPE kit or a new Arima SV detection assay kit, the QC pipeline is free to you. And the SV pipeline is for deep sequencing analysis of IC data from 50 to 500 million reads. Our SV detection pipeline only requires 100 million reads for SV detection as a minimum baseline. Some people choose to go to 50 million reads. We have examples on our end where we can see identify complex structural variants at 50 million reads, but we recommend a minimum of 100 million. And this is available for, this is included with the purchase of Arima SV detection assay and can be purchased separately if you have Hi-C plus or Hi-C plus FFPE data. So I was going to take you through a live demo of this platform. So I'm going to go ahead and switch to another screen here. And to get started, the way to access the Arima Bioinformatics Platform, you can find that in our main arimagenomics.com website, and you can find it under products under Arima Bioinformatics Platform. In that, you can either log in if you already have an account, or you can create an account if you don't have one today. Creating an account is free. So all we need is your first and last name, your work email, and you create a password. And if you already have a coupon code that's provided to you by one of our tech support or sales team representatives, you can go ahead and put that in here as well. Once you create an account, you can go ahead and log in. And I have my password saved, so login's pretty easy. And what you'll see, a typical user, if you've just created a brand new account, you will already have example data on your screen today. So every new user has example data available to them if they create an account on our Arima SV pipeline, Arima Bioinformatics Platform. And this is SV example data. On this project dashboard, you'll see that everything is organized into projects. You can add new samples within each project over here. You can start new analyses over here, and you can upload. This is another method of uploading more samples here. Furthermore, you can access our help function. We have a new help center. So if you have any questions on the portal or the pipeline or the QC pipeline, you can search for them here, and you could, or if you can't find the answer that you're looking for, you can easily create a support ticket, and we will get back to you with information or help or support as needed within a timely manner. So let's dig into our example data set so you get a sense of what you would see when you run your SV pipeline. Here we already have two sample sets, a shallow sequencing sample and a deep sequencing sample. As I mentioned, shallow seq data we do provide for free. All you need is you could reach out to Arima and get credits to get shallow seq sample sets added to your account to be able to upload shallow seq data. From the analysis perspective, we can jump into what a shallow seq data set looks like. And here, when you run our Arima SV QC pipeline, you'll get some basic quality control metrics that are useful for determining whether to move forward in your experiment or if you need to make adjustments from your sample perspective. So your quality score is given here. This is based off of Illumina Q30 scores. You get your alignment metrics and then the QC metrics. And Sophia is going to dig deeper and provide you more context on what types of metrics you would expect to see and which ones we use for pass-fail criteria. Going back, we also have our deep sequencing data set. So in here, we now have the same quality control metrics, the quality score, the alignment, and the QC metrics, but now you have access to structural variant calls and related data. So on the structural variant section, you have a Circos plot that represents all of the structural variants identified in that data set. You have also a table of breakpoints, which can be exported into CSV. So the Circos plot can be exported as an image or an SVG file. So if you have a certain SVs that you have identified that you want to focus in on, you can select them in your table here, and it'll visualize it on the Circos plot. And you can either export the CSV or you can export specifically that image. Furthermore, underneath this data set, you have your Hi-C heat map. which represents everything, all your structural variants and your high-C data visually on a typical juicer heat map. If you wanted to zoom in on a specific SV, you can click on one and it will load in the heat map here. And this is a representation of one structural variant call. And again, Sophia is going to dig deeper into how to interpret the high-C heat map and how to kind of make sense of the data that's generated from here. And also some more advanced analyses options that you can do if you wanted to export the data. So in addition to that, we also have representation of the data in IGV. So I'm going to reset the data here. So that way you can see what the IGV data looks like. It'll just take a second to load. But typically you would see all the data represented. Yeah, here we go. So you'd see the IGV data as a more traditional two-dimensional view of reads within your sample type. So going back to the presentation, we've gone over the Arima Bioinformatics Platform. We had a quick view of the Arima SV QC pipeline and the Arima SV pipeline. I just want to mention again that creating an account on the bioinformatics platform is free to anyone with a valid email address. An organizational email address is all required. Access to our QC pipeline is free for all Arima customers who purchase any of these following kits: the High-C Plus kit, the High-C Plus FFPE kit, or the Arima SV detection assay. I should also mention, by the way, that our Arima SV pipeline is built specifically for human samples. So if you don't have human samples, unfortunately, the pipeline won't produce valid results for you because it is comparing everything against a human genome data set. And then finally, the Arima SV pipeline itself, the deep sequencing pipeline, is included with the purchase of an Arima SV detection assay, where we can purchase it separately from one of our sales team members. And with that, I'm going to hand it back to Sophia, and then at the end of the presentation, we can go over any questions that you may have.
Thank you, Brahim. That was great. We can jump into looking into the report a little bit deeper. So we saw the different sections that Ibrahim demonstrated for us. So I just wanted to mention a little bit more deeply what these sections represent. So the quality scores is the traditional Illumina quality score, the Q30, right? Is that what you mostly want to see for your reads? So as long as your reads are in the green, maybe, yellow region towards the end of the read, you're fine. Ideally, this would be above, let's say, 90%. The alignment section gives you information about your reads, how they aligned on the genome. So we have the uniquely aligned reads. These are the ones that are used basically for all of the downstream analysis. So all the reads that aligned on a specific area on the genome are maintained. There will be some reads that align in different areas of the genome. That is because we have a lot of repetition in the human genome, but because these reads cannot be positioned exclusively on the genome, they're removed from the analysis. And you will always have some unaligned reads, hopefully a small percentage. If the unaligned reads are way too high, you may have some type of contamination in your sample, and you might want to consider taking a look at it. Some QC metrics that are very important for our pipelines. Here in this table, we have some basic statistics that are about your general assay, and also some High-C specific data features that are very important, and some features about your SVs. So if you are working with shallow sequencing data, the metric that you would like to focus on is the percentage of intra-chromosomal reads that are at a distance larger than 15 kilobases. We call these the long cis reads. These are very important because we believe that these are the reads that are giving you the looping and folding information. So we want this percentage to be more than 25% for a pass on your High-C quality control report. And if you're interested in our SV pipeline, then the metric that we're focusing on is the ratio between long cis and trans interactions. These are the long intrachromosomal versus the interchromosomal interactions. We want them to be above 1 to consider your sample to have passed for the SV pipeline. We also have the SV calls that you can view either at a circus plot like Ibrahim showed you, or at the table format. We're going to dig deep into the table, so we can move a little faster here. And we also have the heat map. But another very important aspect is, IGV, if you want to visualize your data this way. And we can move into data interpretation, where I'm going to be showing you a few things on the platform and also locally on my computer. Excuse me for that. I don't have my password saved, so this is taking us a second. So this is the report that Ibrahim shared with you earlier, right? We have all of our data here, and a very important tab if you want to dig deeper into your data or do additional analysis is the info tab. So here you can download every single file that you might want to, right? We have the bedpe file, which is the file that shows you your SV calls, the table that we saw earlier, and the .hic file. So let's say you've downloaded these files and you want to look into your data a little bit more deeply. So before we jump into our juice box, let's talk a little bit about how heat maps look like in the SV context and what do they mean, because they can be a little intimidating at first. So here we have chromosome A and chromosome B, and we see that we have an enrichment of signal that we wouldn't expect before. This is the background right here, and this is the signal that you get between these particular chromosomes, and that is an SV, a structural variant. And also, if you can see this tiny black dot here, our pipeline has annotated this as an SV. So what does this mean specifically? So if you draw some lines from the SV area towards your chromosomes, you're going to be seeing what we call a breakpoint. So the genomic coordinate where, let's say, the virtual line from the SV cuts your chromosome, this is where we have the break on this chromosome. And what does this mean for the actual genome organization inside the nucleus? It means that this part of chromosome B, the top part, and the left part of chromosome A, we see a lot of interactions between them, right? So that means that those two have fused into a new chromosome. This is an interchromosomal translocation. Another interesting type of interchromosomal translocations that I want to mention is the reciprocal. You might see a butterfly pattern or a bow tie pattern sometimes in your heat maps. What does this mean biologically? Again, we draw our virtual lines, we see where we cut our chromosomes, and what this means is that not only the top part of chromosome B and the left part of chromosome A are interacting, but we see a lot of interactions between the bottom part of chromosome B and the right part of chromosome A. So that basically means that these two chromosomes broke off at some specific breakpoints, and then they fused back together, but this time we got two fused chromosomes, right? This is just a way that I'm thinking about, heat maps. Whenever I look at them, I always try to figure out which part of the chromosome A has fused with which part of chromosome B, or in the case of an intrachromosomal translocation, where is the breakpoints of the deletion or the inversion. So generally, this is the reciprocal translocation that we saw. This is the interchromosomal translocation, a more simplified schematic. So there are different types of SVs that can be identified in Hi-C heat maps, and I'm just going to show you two little examples of how a deletion looks like on a heat map. So in this case, this dark purple area has been deleted, and that shows on a Hi-C heat map as some interactions off the diagonal between these light purple regions. And in an inversion, in the intrachromosomal setting, you will see a similar butterfly motif, but it will be at a different direction than the interchromosomal translocation that we saw before. And generally, all the algorithms that are using Hi-C data to identify SVs will, one way or the other, are looking for these motifs. So Hi-C Breakfinder, which is the algorithm that we're using, is in its way trying to identify these motifs. There are some other methods that have been published recently. A very interesting paper that came out was Eaglesea. They use deep learning, but we've decided to use Hi-C Breakfinder because it's more widely supported for folks that either come from industry or academia, right? So how does HiC Break Finder output the data? Here, I want to dig deeper into the table that we saw earlier in the Arima Bioinformatics Platform website. So I want you to focus on the first line, which tells us what each column is, and let's look at the second line. So here I've zoomed into this particular SV between chromosomes 3 and 10, and let's look at the different columns that we have here. So the first column will always be your chromosome 1. And just be careful because sometimes it might be flipped and chromosome 1 might be the one on the y-axis, but usually most of the times the smaller chromosome would be on your x-axis. Smaller in terms of name, not size. So chromosome 1 is right here. And the next two columns represent the coordinates of this square that you see. So HiC Break Finder annotates SVs in the form of a square. That's why we have a BedP file, because that gives you, it's like a loop, right? But it's for SVs, it's the same idea. So you will always have the start of your SV and the end. And also you will have the second chromosome. In our case, it's the Y chromosome, Y-axis chromosome, with the start of the end and the start and the end of your SV. So this table is not as intimidating as it might look. It just tells you the genomic coordinates of the breakpoint on one chromosome and the other chromosome. And HiC Break Finder gives you different resolutions. So here it made this call at 1 KB resolution, but the window, might be of a varying size based on how strong the signal is. So basically here what the code is telling us is that the breakpoint is somewhere in between this area in chromosome 1 and somewhere in between this area on chromosome 2. Another interesting aspect of the way the data is represented is these two columns that are called strands, strand 1 and strand 2, and that basically gives you information on the directionality of the signal. So here, if we imagine that we split this in 4 quadrants, we can have different directions that the SV is going, and that is telling us which parts of the chromosomes are fused. So here we can see that if we draw our hypothetical lines from the end of the signal to chromosome 2, right? This is where the breakpoint is, and same for chromosome 1. And we can see that the direction of the signal is going from the higher genomic coordinate to the lower genomic coordinate on chromosome 1. And similarly, it goes from the high genomic coordinate to the low genomic coordinate on chromosome 2. And that is why the strand is annotated as ++. That's what we see here on the first line of our table. So ++ means that your SV is going from the high coordinate to the low. And I have a little cheat sheet that I've made for myself every time I look at an SV, just to know which corner is the one with the enriched signal whenever you're looking at these signs for the strand. So we saw the info tab that you can go on our platform and download the data. I just want to briefly mention which part of our pipeline generates which file. So Hiccup, which is our alignment and filtering tool, will generate these first files, the BAM files. HiC Break Finder is the one that's generating our breakpoint calls that we saw, which is the BedP file, and it also has it in a TXT format if you prefer. And then Juicer will be the part of our pipeline that generates the .hic file. So after we've downloaded these files, we can take a look at some of the SVs in this particular sample and have some fun. So here I went into the Arima Bioinformatics Platform portal. I downloaded my .hic file, which is my heat map. And I also downloaded the BedP files. So here, this is the Juicebox environment. This is the desktop application of Juicer, which is what we have incorporated into the Arima Bioinformatics Platform. We have the online, let's say, processing environment. But Juicebox, which is something you can download on your desktop, gives you a little bit more flexibility if you want to analyze your data in more detail. And you can add annotations. Here I have added a coverage track we're going to take a look at, and the reference genome, and some oncogenes that I'm very interested in. These are all in the form of a BedP file. If you want to load some genes that you really care about, you can load them here as a bed file. And in the 2D annotations, you can load your BedP file, which is your SV coordinates. Okay, so let's go back to that chromosome pair that I showed earlier, which was 3 and 10. We know here we're expecting a really beautiful translocation. We can see it here. I like to enable my straight edges on JuiceBox so that I can see exactly where I'm going and what I'm looking at. And it's pretty cool here because we can zoom into this SV and take a look at its features better. So every software is not perfect, right? So HiC Breakfinder might give you some false positive calls. So not all the calls you might see on our Arima Bioinformatics platform might be real SVs. And not to worry, we can always review them right here. And what are the characteristics of a true positive, a real structural variant, the way we have been seeing them at Arima? So we want to see very clean edges, right? So you can determine where your breakpoint is with accuracy. So if you see clean edges, that is a plus. And another very interesting aspect is what we call distance decay signal. So I don't want you to get confused. So here we're let's say, zooming into this particular SV. We can zoom in as much as we want. And to verify whether our signal drops in a nice fashion away from the breakpoint, I like to come here and play with the color. So if I keep saturating my color, I'm expecting to come at a situation where only a few bins have really strong signal, and that's where my breakpoint is. So if you are observing this behavior as you saturate the color, and you can see 1 or 2 bins that are clearly stronger than the others, that is a very good indication that you have a real structural variant. And also that bin that is really strong is probably your breakpoint. So what happens when you have a false positive case? So that can also occur. No software is perfect. We are working internally on writing better algorithms to prevent that, but there can be some cases where you get some false positive calls. And how do you recognize them? So this is an example. This is chromosome 19, and we see that Breakfinder annotated a lot of calls here off the diagonal. I want to remind you that it doesn't mean here that we have 10 calls. We actually have 5, but because this is the intrachromosomal space, the matrix is symmetrical, so we do get them twice. If that is annoying to you, you can hide them. I didn't hide them. Here, you can hide them from one end of the heat map, or choose your favorite side of the heat map. So let's zoom in and see why these are false positives. Because if you look here on the side where we don't have the squares, you can kind of see some signal that is enriched. However, as we zoom in to this area, I want you to keep in mind of all the things that we talked about. And it's, what am I looking for? I'm looking for very crisp edges. Do I have very crisp edges here? Not really. And if I play with the color, do I see a distance decay, or is the signal dropping all at once? Looks like the signal is dropping all at once. So this is not a real SV. And in this case, we're calling it a compartment false positive. Because if you go at the chromosome view, you can see that this is probably coinciding with where a normal compartment would fall. So this is an example of a false positive that we have found, just to keep in mind that you might see some of these in your analysis. And the last example that I want to show you is another type of false positive, which is called a low mappability false positive. And let me see, is it here? Okay, I forgot where I put this false positive. I'm really sorry, just never mind. Let me see if I have it in my presentation. Yes, so another type of false positive calls that you can have is in the case of normal structures. So TADs And loops might seem a little suspicious to any program that just looks for enrichment of signal, compared to the background. So what we have here is some cases from some of our samples that we noticed that Hi-C Breakfinder made some mistakes in our opinion, and it called the normal TADs and loops as a SV. So keep in mind that that might also happen. And another interesting case is in areas of low mappability. So as you can see here, we have some regions that are empty. These are most of the times the centromere of the chromosome, and that's because centromeres and telomeres are highly repetitive, and most alignment tools do not align reads there. And that's the case for our Hi-C analysis. So sometimes this lack of signal here kind of triggers the algorithm into thinking that there is an SV. So you might notice cases where you have false positives spanning a low mappability area. But overall, I want to remind you how a true positive call looks like. We looked at this juice box. We want nice clean edges and a signal that drops as you move away from the breakpoint. And you might wonder, why do you want the signal to drop, right? So the way I see it is that you have two chromosomes that fused, like we saw before. You have chromosome A and chromosome B, and this is now a new chromosome. So you would expect that the frequency of interactions to drop the same way as it drops on a normal chromosome, which is a lot of interactions close to the diagonal of this new chromosome, and then interactions that drop as you move further away. So you would expect to see this drop the same way you see it in the intrachromosomal space. So yeah, that's all I have for today. I really want to thank you for being here. I hope the Juicebox tutorial was informative, and yeah, I'll give it back to you, Janetta, to close us off. Thank you.
Thank you to both speakers. Thank you to Sophia for leading us through the introduction to why SVs matter in cancer, that wonderful tutorial as well on identifying true and false positives. I know it can seem really intimidating when you're learning something new, but it actually is not as scary as you think. Ibrahim, thank you very much for the introduction to the Arima Bioinformatics Platform. Okay, so jumping into the Q&A, we have 17 questions. So I just want to remind everyone to upvote the questions that you like. The currently most upvoted question is from Nuet, and the question is, how would this pipeline distinguish reads spanning the breakpoint versus reads spanning TADs? Does the knowledge of the breakpoint have to be pre-existing, or is it a de novo discovery of the breakpoint?
Yes, I can take that question. It is a de novo discovery of the breakpoint. The pipeline does not need prior knowledge. It can identify whatever it thinks it's a breakpoint. That's an excellent question. Sometimes it makes mistakes. Sometimes it thinks that a TAD is a breakpoint. That's why I suggest, if you're really interested in a particular area, or if you just want to review all your calls, just go into the heat map, take a look. And if you think that it's probably a TAD, then it might not be a real SV. This is definitely a field of ongoing research, like how do you make an algorithm identify and distinguish normal structures from a real breakpoint? But the pipeline does not need any prior knowledge. You can just run your Hi-C data. Most of the time it's correct. Sometimes it might have some false positive calls like TADs, but you can just run it and see what happens.
Great, thank you. We're just going to kind of go quickly through the questions, see what we can get through in the next 10 minutes. So another popular question from Ian, is there a size limit for the SVs that you can detect?
No, the only, let's say, limit is that this particular pipeline goes up to 1 KB for the resolution of your breakpoint. So the size of the SV can be variable. The question, I guess, is how accurate you can be with your breakpoint. So currently we can do 1 KB with this pipeline, and we're actually working on dropping that even more.
I also want to mention, outside for intrachromosomal breakpoints, the size of the breakpoint is a lot that we can get down to quite a small size. The pipeline is more likely to call a false positive in the intrachromosomal space, because you're also dealing with natural structures and formations that it has to kind of be able to kind of see through. So there is some variability there between inter and intrachromosomal breakpoint sizes. But the larger the breakpoint actually is, the easier for us to see.
All right. We have a question from Alum. If we have a mutant set with a big inversion in one chromosome, what is the difference between aligning the high C to a wild type reference genome and a mutated one?
Yes, very good question. So most pipelines, including ours, will align your reads to the reference genome. That's why you're expecting to see all these interesting patterns off the diagonal. Because if you had your mutant reference genome already pre-assembled, then you could basically align your reads and you wouldn't see these weird structures, because in that particular genome, it would make sense. So it would be like a normal chromatin structure. That is a very good point. There are some tools that are trying to assemble the genome on the fly in this particular area and align the reads, but in our case, we're aligning it to the reference genome and we're looking for these interesting structures off the diagonal.
Thank you, Sophia. All right, we have a long question from Chaitali. Sophia, thank you for the seminar. I agree, thank you. Do you guys think that loops called and categorized as interchromosomal loops by loop calling tools can be inferred as a bona fide translocational interaction output? Or is it possible that the two chromosomes at that particular snapshot of time and stage were in close proximity? So how do you segregate a true gene fusion from temporal interchromosomal interactions? And you guys really like to ask difficult questions.
Yeah, these are all very insightful questions. And if you have ideas, come work for us. We can develop some algorithms. So
there you go.
This is an excellent question. What I think is that if the interactions between chromosomes are just the normal proximity, right, interactions, then they wouldn't be very strong in the signal. I think those are the ones comprising the background that we see, because the interchromosomal space is not empty. It has some signal, but we think that that's the normal frequency of interactions due to just proximity. Usually when there's a breakpoint and there are a lot of these new loops spanning the two chromosomes, the signal is really strong.
Thank you. All right, moving on, question from Jack, or I guess two questions. One, can long read sequencing partially solve the low mappability issue around centromeres and telomeres? And two, is there any way to find SVs during genome assembly, or do we need a reference first?
I could take a part of that, Sophia, if you wanted to answer the first one or the second one. But I think for both of these, coupling high C with long reads is definitely going to give you higher quality data sets. One of the advantages that we like to talk about for high C data is, when you're working with like FFPE or clinical samples, there's a huge advantage there, right? Because long read sequencing data is not able to work with that as well. You don't get the advantages of long read sequencing because you have damaged DNA and sample types. But if you have fresh samples, I think it just can yield to better research and better, data together by coupling two really useful and valuable technologies together to kind of give you a better picture. And that's why in genome assembly projects, you typically do combine high C data with long read sequencing data. And then on the second one for, is there any way to find SVs during genome assembly? We do need a reference. So high C break on our SV pipeline that's hosted online, we have it built, baked in with the human genome reference data set. But the high C break finder pipeline uses something called an expectation file, which is your reference genome. And that expectation file is how the high C break finder is able to kind of try to determine as best as it can what's the breakpoint versus a general structure. So there's that component that you need to have some sort of reference to use with high C break finder.
And that's a digest file that's, I think, produced. I'm just looking at the GitHub on our page. It's a genome digest file produced by HiCub Digester. So I suppose if somebody has a bioinformatician or knows how to run things from the command line, they'd probably be able to create that digest file and add it to the pipeline. But I'm not a bioinformatician, so with a grain of salt.
Yeah. Anything to add there, Sophia?
No, I think your answer was perfect.
Yeah.
Great.
I could do it sometimes.
All right. Okay, we have a question from Naoki. the question is, do structural variants tend to occur randomly, or do they have specific patterns and can be classified in patterns?
So I see two sides of this question. The first side of whether they can occur randomly, I think yes. There is definitely research going on to determine whether some specific chromatin features might make a region more prone to an SV, like a breakpoint. I'm not aware of any specific features right now, but the second part of the question about the patterns, I think most SVs will, let's say, manifest themselves in either the butterfly pattern that we saw or some like, let's say, signal of to one quadrant, right? So there are patterns on the high C heat maps that signify that an SV was there, and you can identify the exact type of the SV from the specific pattern on the heat map. But whether the SV has is primed to happen in a specific area of the genome, I think that's an amazing question, and it's definitely not resolved, right? I think I understood the question correctly.
We do have like 2 minutes left, so I would like the speakers to just briefly scroll through all of the questions that that you currently see and tell me, is there a question that you think is really good that you would like to answer? And let's see if there were any other questions. Yeah, there were some questions about other genomes besides human. So the SV pipeline that Arima has in the Singularity image, as well as on the Bioinformatics platform, only is compatible with human. And if you do have bioinformatics expertise or can have access to a bioinformatician, you'd need to create your own digest files.
That's right. I was just going to say there's a decent number of questions around, changing out the reference genome, and all of that is certainly possible. It's just not something that is a baked-in feature to our pre-built pipelines. But that's also why we use open source pipelines. We want to work with pipelines that enable research, and that allows you to kind of get you at least a large portion of the way. You can open it up, and then if you have access to the expertise, kind of swap out things that need to be swapped out to make it more applicable to your research. Our hope is to kind of streamline the research for as many people as possible. And in this particular case, we're really trying to streamline the research for human genome structural variant calling.
Just one last comment from me. I see some questions about deletions or duplications, and I know during the talk, we mostly focused on translocations, but the pipeline does identify deletions and duplications. It doesn't classify them as of now, like it won't tell you this is a deletion, but by reviewing the heat map, you can, infer that.
And one last question I wanted to address from Jason, Jason Howard, for your PDL1 gene fusion detection, how many PE iSeries did you run? Simple question. It's a good question. Actually, we saw this in our shallow-seq data set. So the first time we noticed the PDL1 gene fusion was on a 15 million kind of shallow-seq run. But then, our collaborator, Dr. Sundaro, actually wanted a deep sequencing data set because he was actually interested in the more of the regulatory aspect of the sample prior to us finding the PDL1 gene fusion. So we have both the shallow-seq data set and then the deep sequencing data set. So we can definitely see these variants and abnormalities occur in a shallow-seq data set, but the reliability of those finds can vary depending on the size of the break and other factors. In this case, it was just a shallow-seq set that allowed us to see that one this time.
So many good questions.
Yeah. There are so many good questions. I know.
So many good questions that
maybe we should have like a webinar where we just like go through answers to these questions. But yeah, thank you all. We are over time, want to be respectful of your time and meetings and so on. If there are questions that we didn't get to that you really would like an answer to, you can always email us, team@arimagenomics.com. Otherwise, thank you so much for joining us, giving us an hour of your time. We hope you're energized and excited about structural variants. And thank you to both of our speakers, Sophia and Ibrahim, for preparing and spending a lot of time to put on such a fantastic webinar. Thank you, everyone.
Thank you.
Bye.
Thank you.